Thank you for attending this last lecture in the series of the International Francqui Professor for 2021 and 22 at University of Antwerp. We will continue to focus on the weak newborns, both infants and animals, and this time have a specific focus on microbial interventions that we can do to improve the health of weak newborns. Quick introduction to our group in Copenhagen that work with these areas. And as I mentioned, we will focus on the immature and small newborn infants and immature and small newborn piglets and translates between the species. So feeding and colonization transition at birth. What is happening at birth? Where we have the most dramatic physiological change in the life of mammals. When we come from the sterile parenteral nutrition condition in utero before birth to a colonized state of enteral nutrition after birth, we have to decrease parenteral nutrition and the sterile state and increase the state with enteral nutrition and bacteria. So the bacteria per gram of feces in this time increases dramatically in just a few days from basically sterile gut to 10 to the 10th bacteria per gram of feces. And there's a great variation from 10 to 100 different bacterial species in this time period. So this is a very dramatic transition in this first week or so in all newborn mammals. When we would want this transition to a colonized gut to happen is a question mark, and the degree to which we would use antibiotics, who would we treat it, when would we treat it, and what would we give is also a big question, especially when it comes to the very weak newborns. We can also question whether we would give this antibiotic systemically or by oral route, and whether we would modify the colonization that we are trying to achieve with pro and prebiotics. And then comes in here which product to use, which dose to use, and when to use it. Finally, all these things also interact with a different diet that we may use during this important transition period, whether we would use a formula diet, mother's milk, or milk from another species, or donor milk. So many questions. First, we'll take a look at birth, what it actually does to gut microbiology, and also to the sensitivity of gut inflammation, NEC, that is necrotizing enterocolitis, a very serious gut disease in immature newborns. So NEC looks like this in infants, and it is basically a very serious gut inflammation that is very dependent on the gut microbiota. So it is adverse feeding and gut microbiota effects to the immature gut. How does different delivery methods influence the gut microbiology and the NEC sensitivity? We did some studies in preterm pigs many years ago where we compared cesarean delivery and vaginal delivery even within the same litter. So delivering the first half of a sow litter with cesarean and then giving birth to the remaining piglets by vaginal birth 1 day later. What we saw for the microbiota is as expected on day 5 here, that the microbiota is very low density and with few species when we have cesarean section. But we had about a 50% NEC incidence in those that were cesarean delivered. If we compared that to those that are now vaginally delivered, which would have the chance to suck up mother's own vaginal flora, we have a much more diverse microbiota in the gut of these newborns 5 days later, which we would expect, and the density is higher, but the NEC incidence was actually the same. So it seems like some of the benefits and advantages of colonization are balanced out when it comes to delivery method. And that is exactly what we see also from clinical practice of infants, that the incidence of gut inflammation is not any different after a cesarean versus a vaginal birth, even though the gut colonization is vastly different. So it's not only gut colonization that determines the degree to which we may experience gut inflammation in these sensitive newborns. Gut permeability is a problem in the immature newborns and also in the small newborns. So here is a study that compared 116 preterm infants with 16 term infants, and we saw that the gut permeability as assessed by the lactulose mannitol ratio was significantly reduced in the term infants relative to the preterm infants. When we have a high gut permeability, there is a risk that these bacteria may translocate across to the bloodstream, both the bacteria themselves and their toxins, which may create bacterial translocation to the bloodstream and systemic inflammation, even to distant organs like the brain. So the barrier problem after preterm birth, we will just highlight it a little further. When we have the state of birth, we go into a world of microbes where we have 3 different epithelial layers that need to protect us from invasion of bacteria. These layers are the skin, the lungs, and the gut. And when we have a state of healthy development, then these bacteria do not go across to the bloodstream and to the brain, of course. Some factors go across and other factors are inhibited by the skin, lung, and gut barrier, and other factors are then inhibited across the blood-brain barrier. So we have a protected system in a healthy environment. Conversely, if we have immature barriers in compromised newborns, bacteria and their toxins may cross the gut barrier and even the brain barrier, the blood-brain barrier, which will create a whole series of brain inflammation conditions that I just mentioned here by their abbreviations. So this is very commonly known for preterm infants that have immature barriers, that they have very frequently brain inflammation disorders. When we have immature barriers, we will have a risk of infection and inflammation in the bloodstream, potentially leading to late onset sepsis and late onset bacteremia in these immature or small, weak newborns. This may also create longer term immune defects, and these immune defects may spread to diverse range of organs around the body, not only the brain, as I mentioned before, but also the kidney, the bone, and the muscle, and not the least also the gut, so that the systemic inflammation may also go back and actually thereby further enhance the inflammation that was starting in the gut. I would like to show you how we can locate the bacteria close to the intestinal villi and how that is particularly a big problem in the preterm newborns. So what I show you here is a cross-section of an intestinal, small intestine from a preterm pig, where we see all the bacteria marked with the red color here, and you see how closely they are associated with the mucosal lining. And this is a specific feature of preterm newborns, is that the bacteria, they get too close to the mucosal lining. So we can also mark these mucosal linings and quantify the degree to which the bacteria are closely associated with the mucosa. So here, that is clearly an indication that there is a problem, because the mucus peria doesn't really protect these red colored bacteria from interacting with the mucosa. If we take a closer look, you can also see the bacteria here with the red color interacting very closely with the villus surface, which is generally considered a problem and creating inflammatory reactions. So we can quantify these interactions and then see the effect of different interventions. Now, let me show you here the intensity of this epithelial bacterial contact in a cross-section of an intestine. Again, the bacteria is marked here in red, and this is again the cross-section of an intestine from a preterm pig used as a model for an immature infant. So the factors that influence the bacteria in the gut lumen is first and foremost the feeding. So the feeding will stimulate gut motility and will inhibit the bacteria from attaching to the mucosal lining. So this means that feeding is important to avoid bacterial attachment to the mucosal lining. Constipation, dysmotility, and ileus of the intestine is a common feature in immature newborns. And when we have constipation and dysmotility, the bacteria has a chance to interact closely with the bacterial lining and also to proliferate, because they are not being transported down the intestine to the same degree. So ileus and constipation is a problem also in these infants due to their dysregulated motility. A formula diet without any protective factors or antibiotic-like factors in that milk will not inhibit then the bacteria from proliferating, and will not also inhibit the bacteria from interacting closely with the mucosal lining, and may also then induce that the bacteria transverse into the mucosa. A milk diet with protective factors, conversely, will inhibit the bacteria from close interaction with the epithelium, which may be like an intact milk or colostrum diet relative to a formula diet. We may also be able to control the microbiota via probiotics and prebiotics. And what is it probiotics and prebiotics are doing in such a sensitive intestine? Well, by competitive actions to other bacteria, they may inhibit the adhesion of the pathogens to the mucosal lining. So there's many ways whereby probiotics and prebiotics may interact and stimulate the mucosal defense, and I'll come back to that in a minute. But finally, we need to mention also antibiotics treatment that is done often to also decrease the exposure of the mucosa to bacteria. And of course, antibiotics will reduce the number of bacteria that are interacting with the mucosa and thereby also inhibiting the inflammatory reactions. Let's get back to those three different ways of manipulating the bacterial contact with the mucosa: prebiotics, probiotics, and antibiotics. And this will give us a picture of how we can potentially manipulate the bacterial colonization to be in favor for the health of the newborn. So first we'll take a look into human milk oligosaccharides, which is basically prebiotics derived from milk. And here is a paper that was led by Stine Bering, where she summarized some of the studies that we have done over the years, also based on newborn, term, and preterm pig studies. And what we found generally was that the term gut interacts quite positively with prebiotics from milk, these human milk oligosaccharides. And in a term gut, we have a well-functioning peristalsis that will allow the bacteria to bind with the human milk oligosaccharides. And the human milk oligosaccharides will also increase the microbial metabolite production and stimulate the restitution of the enterocytes and create an anti-inflammatory state via certain cytokines such as TGF-beta and IL-10. So this is what we found for the term infants and also term newborn pigs. But if we compare that situation with those more sensitive preterm newborns, it may look something like this. So we have a reduced peristalsis of the gut that reduces the activity and the motility of the intestine to interact closely with the feed and progress the feeding down the gut. This may lead to pathogen inhibition to receptors on the intestine because the intestine is not moving right, which will also allow then pathogen proliferation in the intestine and colon, mucosal invasion by bacteria, and reduced restitution of the enterocytes in this condition, potentially leading then to inflammatory cytokines being released in the submucosa. And these HMOs may, in the worst case scenarios, also actually be affecting both the brain and the urine, but to a very varying degree in preterms relative to the term newborn gut. So this is just an overall picture of how there may be differences in the way that human milk oligosaccharides, the prebiotics of milk, may interact with the gut in the term versus the preterm newborn. So HMOs are not as effective in immature infants. That is our conclusion. Now, is early colonization really important for weak newborns? Well, we may ask that question because the intestine is, of course, immature, and so is its immune system. Its immune system is very determining for the development of a disease like necrotizing enterocolitis that we can use as a landmark for adverse gut effects in the immature newborn. So here I just report a Cochrane collaboration review on all the probiotic studies that have been done to prevent necrotizing enterocolitis in preterm infants. And this is absolutely impressive. So in the last 15 years, there is nothing less than 56 recent studies that have studied NEC in response to probiotics. And generally, the NEC incidence is decreased to about half, but the sepsis is not affected. But what I want to show you here is also what are a huge number of studies that are required for us to inform clinical practice. So is then probiotics being used in clinical practice for preterm infants? No, not yet. And that is amazing. After 56 studies, we are still confused about whether we should use it or not use it. And here I just show you an outline of all these different studies from the Cochrane review here. And you see generally the tendency to reduced incidence of NEC in this forest plot, although not a super big effect. So the question is still remaining, which infants should we treat? Is it only the very weakest infants, or are the weakest infants also receptive to maybe detrimental effects of overfeeding with probiotics? When should we give the probiotics? Should we give it straight from birth or a few days later together with enteral feeding? Which dose should we give of probiotics? And then finally, not the least, which strain or which strain combination should we give as a probiotics? So you can maybe sense now that there are many questions still remaining, and there are some cases of adverse effects of probiotics that make a lot of units be hesitant to actually use probiotics as a standard routine in the neonatal clinic. Now, we also tried to investigate this, of course, in our preterm pig model, and we had 2 papers in Journal of Nutrition about 10 years ago that was somewhat provocative. Initially, we had a 5-dose combination of probiotics that showed a beneficial effect because the bacterial probiotic combination limited the diet-induced gut dysfunction and necrotizing enterocolitis in these piglets. So all was fine. We were supporting what we were expecting, that probiotics would reduce incidence of NEC. However, a couple of years later, we performed another study with 3 of the strains in a slightly higher dose, and the incidence of necrotizing enterocolitis was increased following this probiotic administration to preterm pigs. So why did that happen, and why could we have achieved such different results? We believe it's mainly due to the slightly higher dose used in the second study, but this also indicates that probiotics may actually potentially be detrimental when we have very immature newborns and are using it in a too high dose. So is this representing too much of a good thing? We certainly believe that there's a need to be cautious. So could we potentially use fecal microbiota transplantation instead of a few different strains, so that we would simply transplant the microbiota from a healthy donor into these immature newborns to try and help their natural colonization? It is also shown that a combination of strains is much better than single strains. So therefore, maybe fecal microbiota transplantation might be a way to prevent NEC, and we are currently studying this both in pigs and infants. But I'll just show you here some of the first results that we came to a few years ago. So what we found that this fecal transplantation of microbiota was beneficial, but only under certain conditions. So here I show you our NEC scoring for the stomach, for the small intestine, and for the colon with increasing severity. And when we gave the fecal transplantation, both orally and rectally, then we had a decreased survival of those that received the FMT, the fecal microbiota transplantation. So they died of sepsis. And that is, of course, what you fear when you give bacteria to these infants, that you may have a detrimental effect like sepsis. The motor activity was significantly also reduced, indicating their poor health outcome. Their growth rate was decreased, and the NEC incidence was lowered in the FMT group in the remaining pigs, in those pigs that remained until the end of the experiment. So the treatment helped to reduce NEC, but a lot of the animals actually died before they got there. So that is, of course, a problem. Now we changed to give the fecal microbiota transplantation only by rectal route, because potentially the stomach and the foregut is not designed to tolerate such high amounts of bacteria, especially not in a preterm newborn that has a very immature immune system and immature gut motility. Survival rate in this experiment was the same in the two groups. Motor activity was the same, and the growth rate was the same, and now positive for growth both groups, and still a significant reduction in the NEC incidence, and there was no pigs dying. So this is indication that the route of administration is very important also for trying to manipulate the gut microbiota. So the conclusion is that fecal microbiota transplantation may help, but it is dangerous, especially if you give it via the oral route. So can we schematically show it a little bit, what we want to do in these immature newborn guts? When we have an okay bacterial tolerance, it may look like this. We have the microbiota coming in from the oral side, and then the microbiota goes through the gut. But we may also have microbiota come from the fecal side, from the rectum, and traveling into the gut. And we actually believe that the natural colonization comes both ways. It's not only by oral route, but it's certainly also by rectal route. And in this case, we have typically development of a beneficial fermentation, immune maturation, and protective acid barrier that helps this gut to function normally. So this is when the bacterial tolerance is okay. Now let's, and in this case, we would keep on colonizing for digestive and immune maturation, even by giving probiotics. What then if we have an immature motility and immunity in the gut? Then the situation may look like this. If we compare it again to the previous situation, we may have microbes coming in, but they get stuck due to dysmotility. And we have microbes coming in from the rear end, and again, they also get stuck. The gastric acidification may not be sufficient because we have an immature acid barrier in the stomach. We may still achieve a maturation of immunity and motility by giving probiotics. And we may have a moderate nutrient fermentation if we have this sort of moderate state. So in this, what I wanted to show here is the moderate state of immaturity in motility and immunity. So in that state, it may still be beneficial to give the microbes and to give probiotics if we can balance it right, not give too high a dose too early, but together with a protective diet, then this should actually be beneficial. And that is also indicated from many studies that if we give it together with mother's milk, with gradual feeding approaches, then probiotics is beneficial. But now let me try and illustrate when I think probiotics can potentially be harmful. So in this situation, we would keep on colonizing with good bacteria. So if we give too much and too early for colonization in an immature gut, how may it look like? Microbiota may come in from the the oral side and from the rear end, and we may have a very fast passage and diarrhea, or we may have an ileus condition that allows then the microbes to ferment and colonize, and we may get an over-fermentation of both metabolites and microbes, and bacterial overgrowth, which will give inflammation, and then we have the problem arising, bacterial overgrowth and inflammation, and a negative feedback to the stomach, and then have a poor return flux of, so this will give gastric residuals and vomiting for that infant. So this is a situation where we really would not want to add more good bacteria, not even good bacteria. We would rather try to inhibit colonization and prevent that this infant or pig would develop NEC or sepsis. So typically, how would we do that? We would do that with providing antibiotics. So this is, of course, a serious problem. So we can summarize a little bit that the feeding of the newborn infants is a combination between nutrition, microbiota, and immunity, but also it's very important to realize whether we should treat that newborn weak infant or animal as a fetus or as a newborn. If we would treat it like a fetus, we would not tend to stimulate the colonization effects, but be very gradual in both the feeding and the colonization. If we would feed it or colonize it like a normal newborn, we would try and promote the colonization and also the feeding responses. We would want to feed for gut maturation without maldigestion and fermentation. We would also want to give bacteria for immune maturation without getting into a state of bacteremia and inflammation. So this is the very difficult balance that we are facing with these immature newborns. What I want to show you here is how antibiotics is then being used for some of these immature newborns to help prevent excessive fermentation and the microbial stimuli in the gut. So here is a study from Finland on normal term pigs, where the effects of amoxicillin treatment of newborn piglets, just one injection of amoxicillin antibiotics, on the prevalence of hernias and abscesses, growth, and ampicillin resistance of intestinal coliform bacteria in weaned pigs. So this is an interesting study because this is a study of neonatal preventive antibiotics, which we would not normally want to do and encourage the farmers to do. But since there is generally a body weight advantage for doing this antibiotics treatment, here indicated with the green bars, which is highly significant in this study, it is tempting to use this neonatal antibiotics treatment for the farmers. When it comes to the mortality rate, it was also markedly actually reduced in the two antibiotics group, not only in the first week of life, but also across the entire first four weeks. So at the moment in Denmark, about 90% of the farmers, they are giving neonatal antibiotics, 1 or 2 antibiotic doses, even if it's not recommended to give preventive antibiotics. And that may be due to these beneficial growth effects to the animals. So why is it that this neonatal antibiotics is beneficial? Probably it is because it is reducing the density whereby the bacteria, they interact with the mucosal lining of a relatively immature newborn intestine. We also tried to then study this antibiotics in context of NEC resistance and immunity after 5 days of oral antibiotics treatment to pigs fed an artificial formula diet. And this gave us the most dramatic effects to reduce NEC that we have ever experienced. So I'll show you the results here, where we could reduce both the aerobic and anaerobic bacteria by antibiotics treatment down to about 10 to the 5th in the colon contents here. And the NEC incident was completely eliminated when we gave antibiotics for 5 days relative to the control group that had 80% or more than 80% NEC. The NEC severity score was also correspondingly markedly reduced. So a very dramatic effect of giving neonatal antibiotics to reduce NEC. Organic acids in the stomach and colon was also markedly reduced in the stomach and also markedly reduced in the colon. Histology, when we compare the antibiotics treated pigs with the controls, it looks like this, a very dramatic difference. And the villus height was much higher in the antibiotics treated pigs relative to the controls. So a very marked positive health effect of giving neonatal antibiotics. The goblet cell density in the colon region, you see here in the antibiotics group, there is no need for use of these mucins in the goblet cells in the antibiotic treated pigs, whereas in the controls, the goblet cells are empty and have also already released their mucins to try and fight the bacteria. The bacterial load in the blood, the bone marrow, and the spleen is partly related to this mucin production because the mucin protects against translocation. What we find is that both systemic and oral antibiotics here in this experiment reduced the cultures in the blood, also in the bone marrow, whereas in the spleen there was no difference. So why would we have to reduce still, even if it's beneficial, unnecessary antibiotic treatment? There is a series of consequences of antibiotic theory that I'm sure you're all aware of. There's toxic side effects to specific antibiotics. For example, gentamicin has effects on the kidneys. In vitro studies also show different cellular responses to many antibiotic drugs, and there's numerous papers that show various negative effects of antibiotics. So that is one reason to, of course, reduce the amount of antibiotics being used. A secondary reason is that antibiotics create gut dysbiosis. So the antibiotics may affect not only the gut microbes, but also the underlying immune system that will then develop differently in those that have been antibiotics treated. So the gut dysbiosis indirectly then affect the development of the immune system. And I'll get back to that now a little later in a few more slides. How is it that neonatal antibiotics may at the one hand be beneficial and have beneficial health effects for the intestine, and on the other hand have detrimental effects on the development of the immune system? And finally, it's also important to note that there's the antimicrobial resistance that is a problem. So therefore, we cannot, without any reservation, recommend neonatal antibiotics treatment. So the antimicrobial resistance problem is, of course, a big one across both human healthcare and in the animal industry. So gut dysbiosis and immune system interaction there. Here is what antibiotics is doing. This is an overview of how antibiotics induce microbiota changes and affect the immunity. And there's a number of papers that report all these microbiota changes and effects to the immunity. What the antibiotics may do is that they may reduce the antimicrobial defense a little longer. They may reduce the expression of key cytokines, reduce expression of receptors for bacteria, and suppress the immune response to oral vaccination, and finally reduce the bactericidal activity of many organs, including the bone. So how can we experience these short-term benefits of antibiotics when we have all these negative longer-term immune effects? It partly relates to changing of the gut microbiota, but I'll show you some results that illustrate this difficult balance between giving antibiotics for treatment and then the longer-term effects on the immune system. So antibiotics was given to these mice. Here's an experimental study These researchers gave no exposure or short exposure and long exposure of antibiotics. And here long was defined as 7 days of antibiotics versus 3 days of antibiotics in newborn mice. What we saw was that the Clostridia, for example, increased in density when we had the longer-term antibiotics, longer-term exposure here, which indicates that it really makes a big difference how long we treat with antibiotics. Also, the percent of actinobacteria were markedly upregulated after longer-term exposure. The intestine immune response was not really affected unless we had the longer-term exposure, and likewise also the TLR4 to TLR2 bacterial receptor levels was mainly affected when we had the longer-term exposure. And finally, and importantly, we found that the late onset sepsis, the later sepsis in this mouse model of antibiotics treatment was after long exposure of antibiotics markedly reduced the survival here. So mortality greater after longer term of antibiotics treatment. So it's important that we consider the length of the treatment when we want to achieve the benefits for the immature newborns. Here I'll then turn to some blood transcriptome studies that we did in the next sensitive preterm pigs after antibiotics treatment. Again, further to investigate what is it that neonatal antibiotics treatment is doing to the microbiota and the immune system, and how may that be beneficial or detrimental longer term. What we did, we gave the pigs 5 days, so on sort of intermediate length of treatment, and looked at the blood transcriptome on day 9. And this study was done by these 3 important people here. And we studied the genes of the blood side, so the blood genes, and which genes were upregulated and downregulated in the control group versus the antibiotics treated. And you see here the downregulated genes with the blue color, and you see that maybe already here that most of the antibiotics animals are blue in color, indicating that a number of genes are downregulated if we treat with 5 days of antibiotics. But please note that the blood taken for these studies here were actually taken on day 9, 5 days or 4 days after the stop of antibiotic treatment. When we did a pathway enrichment analysis, we found about 1,500 differentially expressed blood genes, and 1,200 of them were downregulated, and much fewer of them were upregulated. So there is an indication that the blood genes related to the immune cells in the bloodstream are downregulated when we treat with antibiotics, which of course would tend to inhibit those blood cells from being able to fight bacterial infection in the bloodstream later. So when we did a lot of other analysis of these transcriptomic pathways, we have indicated here 9 different pathways. We see again and again the same picture, that the AB treated animals over here to the left are having the blue color, which is downregulated, whereas compared to the controls, which has an upregulated expression relative to the antibiotics ones. So it may indicate that antibiotics is depressing the gut microbiota, which we were also investigating in these other studies. We know that already. It reduces NEC and gut inflammation, no doubt about that. But the problem is that it delays also systemic immunity development. So therefore, you may then have a counter strike in infections later when we treat with early antibiotics. Suppression of the immune system may happen by many ways related to the microbiota. The microbiota is changing very much when we have development of inflammation in the gut, and it appears that enterocolitis changes in the gut microbiota may also induce systemic immune suppression in the bloodstream of neonatal preterm pigs. So I show you here some results, some other results from preterm pigs where we documented this. And this was led by Ning from our group. So in this study, we characterized the degree of inflammation in the gut as severe NEC that might potentially lead to bacterial translocation and late onset sepsis, but also subclinical NEC or mild NEC, which would be associated with a decreased systemic immunity and not necessarily development of late onset sepsis. But let me show you how these different states of inflammation of the gut may affect the systemic side, the blood side, and then increase the sensitivity to sepsis later. So I'll just skip this slide. The feeding with bovine milk diets for 5 days, and then blood and tissue collection at day 5 and day 9. And I group here the pigs into no NEC, mild NEC, and severe NEC. And you see here a picture of these colons that were affected very aggressively when we come into the score read in 4 to 6. And what we saw for the NEC incidence was that it was similar between day 5 and day 9, and also that the villus height in the small intestine was similar across the severe NEC and the mild NEC. This indicates that the small intestine was not affected. And when the small intestine is not affected in this systemic inflammation condition, then it's a condition where the inflammation is relatively mild. Because what we have observed is that conventionally, there's only a big time problem if the inflammation and the bacterial colonization and over-fermentation occurs in the small intestine. So in this subclinical NEC situation, we found that on day 5, there was an upregulation of the plasma TNF-alpha level, indicating that initially the animals, they sort of mobilize a capacity to respond to bacteria on the systemic side. So we see here TNF-alpha responses on the systemic side as being a positive response to LPS here that may help the animals to fight a later infection. But later on day 9, this response was decreased, both with the mild NEC and the severe NEC in the colon region. This indicates that a subclinical inflammation with mild lesions in the small intestine may later depress the immune response to fight a bacterial inflammation. This is also indicated here on day 9, where we have an upregulation of the Treg cells, T regulatory cells, that are a sign really of delayed immune maturation. And when we studied the top 10 hop genes of the gene expression analysis, we saw that all these genes were downregulated in the studies on day 9 and after NEC, indicating again that if the intestine have been subjected to a mild subclinical inflammation, that this will inhibit the blood ability to fight later infections. So prolonged mild NEC lesions induce systemic immune suppression. So now I'll just return to the 2 slides that I had to skip before because there was a mix-up of order here. But when it comes to neonatal antibiotics treatment, then we also tried to do this and study this in infants. And I show you here the overview of antibiotics treatment in 13 neonatal intensive care units that we studied over 5 continents some years ago, a total of 3,000 very low birth weight infants. And what is important here is to see that 70 to 90% of all these infants were treated with antibiotics during the first 3 days of life. The question is whether these 3 days of life will be detrimental for their immune system, for their later immune system development along the lines of what we discussed before, or will it also help promote the colonization of the gut in a more healthy way so that you actually depress a little bit the microbiota and have a reduced risk of serious gut diseases like necrotizing enterocolitis. Let me show you an extract of those results. So we had early antibiotics within 3 days of life in about 2,500 infants, and we had an NEC incidence of 4%. In the remaining infants, about 270 infants there, we had 24 of these infants that developed NEC, which is a proportion of 9%. So the incidence of necrotizing enterocolitis was nearly or more than doubled when we did not give neonatal antibiotics. So we would not conventionally want to give neonatal antibiotics, but we do it to these immature newborns because we don't dare to avoid it, and because the newborn may be infected, and it actually seems like it also prevents against NEC. So early AB was associated with less NEC, and the odds ratio was phenomenal. So there's no doubt that this was a true effect of giving neonatal antibiotics that may also reflect the study that I mentioned in pig production in the beginning, where just 1 day of antibiotics treatment had long-term effects on these newborn piglets. So how do we balance the benefits versus the harm of neonatal antibiotics? That is a very hard question. So I'll just skip this slide again that I showed before and show the final slide, which is sort of a summary to try and explain how we should balance to colonize, to promote colonization or inhibit colonization in these very weak newborns. So should we protect or challenge the immature gut and the immune system? That depends on many factors. So we have an immature gut epithelium in the small, weak, and immature newborns that may easily end up in inflammation. And it is an interaction between the microbiota, the nutrition, and the immunity when it comes to avoiding inflammation in this gut. This inflammation in the gut may, however, spread to a number of other organs via the systemic circulation and via systemic inflammation and increased sepsis and bacteremia sensitivity. So if we have a very sensitive newborn that we want to protect, we may want to delay gut colonization with, for example, having very high hygiene and antibiotics treatment. And we may want to use slow feeding advancement to avoid also bacterial over-fermentation. And we would only use a protective diet like mother's milk. On the other hand, if we have a more robust infant or newborn animal, we can justify fast bacterial colonization because the gut can tolerate it. We may even combine it with early and fast feeding progression, and we might even want to consider formula feeding because the gut itself has enough protective barrier to be able to protect itself against any harmful bacteria. So in essence, we want to control the gut and systemic inflammation this way, but it may very much depend on the state of maturity. So we might want to take this pathway when we have the very immature newborns, whether it being animals or infants. And when we have a more moderately or more mature newborn, we can attempt to take the other path if necessary. So this is the difficult balance that we are in when it comes to controlling the gut microbiota. So it's hard to control the microbiota. It's harder than controlling the feeding, and it interacts a lot with the feeding. But we have to try and do it, and therefore this is a very difficult problem for caring for the very weak newborns. So with that, I will end my talk now and open the floor for any questions. Thank you very much for your attention.